Okay, sorry we didn't get a chance to finish the rest of this in class. I'm going to go through it quickly with you right now, and then also the next little section that I'm not there for today. Okay, so we've seen this already. I'm going to flip through. Fiddlesticks. Okay. Oh. So we have talked about. Hold on a second. Okay, so I think this is kind of where we left off. The whole point of the electron transport chain is, well, really to transport electrons from one complex to another. Now, for your test, it's totally fine if you refer to these complexes as 1, 3, and 4. Okay, don't worry about the names. You can call this guy ubiquinone or coenzyme Q, it doesn't matter, and this is your cytochrome C on your template, okay?
What are you talking about?
Okay. All right, so the whole purpose of these cofactors is to shuffle electrons from NADH, okay, from complex 1 to complex 3, then from complex 3 to complex 4. And the movement of those electrons allows those complexes to transport or pump, really, protons into this intermembrane space, so the space between the two phospholipid bilayers of that mitochondrial membrane, okay? There's also, don't forget, complex 2, which on your template was around this area. Complex 2 is stripping protons and then thus electrons from FADH2, okay, converting it to FAD plus, okay, or FADH, either one. So that's kind of where we left off, and remember that now under here is oxygen, okay? Remember that it's oxygen with its super high electronegativity that is kind of pulling the electrons along this chain and in the process reacting with the protons that are in the mitochondrial matrix to form water. So that is where we left off. So what we didn't talk about together in class is what is pulling the electrons. We did talk about oxygen. Oxygen really is the primary electron acceptor with its high electronegativity pulling electrons down the chain. But the order of the complexes is really, really important. The complexes are ordered in terms of their relative electronegativities, okay? So oxygen is more highly electronegative than complex 4, but complex 4 is more highly electronegative than complex 3, which is more highly electronegative than complex 1, okay? So the cofactors that we mentioned, which is the ubiquinone or cofactor Q, and then the cytochrome C, are shuttling the electrons from one protein complex to the other, okay, and the complexes are the transmembrane proteins, okay. So the electrons are essentially moving from carrier to carrier, kind of downhill, okay, to shuffle the electrons along, and it's all based on electronegativity differences between the complexes and then between complex 4 and the oxygen, okay. So this is a picture as well that matches your template. Again, for whatever reason, protein complex 2 is not in there, but remember that's the guy that's kind of dealing with the FADH2, which is another electron carrier. And the electrons are the yellow lines, okay, representing the flow of electrons, which are picked up eventually by the oxygen in the process forming water. And then we're into the actual ATP formation, so that's the goal, okay? The rest is just moving electrons, really, and pumping protons, but then it's that proton gradient that ends up allowing this ATP synthase, okay, which is it's a transmembrane protein, but an enzyme, because remember some membrane proteins can also have enzymatic activity. So this ATP synthase, the name kind of tells you what it's responsible for. It's synthesizing ATP, okay, using that potential difference. Remember, we talked about this a little bit when we were talking about active transport. You can use the concentration gradient of these protons as a source of potential energy, and really it's a electrochemical energy that allows the phosphorylation of ADP to form ATP, okay, by using that electrochemical gradient.
Mommy.
Okay, so that's what I was talking about here, the gradient, okay. The protons are going to diffuse from that area of high concentration to low concentration, so don't get confused. This is not active transport. You do have a concentration gradient, and it is diffusing down its concentration gradient, okay? And it diffuses, the protons diffuse down through this ATP synthase, which allows the formation of ATP. Okay, the mechanism is described a little bit in your textbook, so you can look at that. What these arrows are representing here is that the ATP synthase actually kind of rotates. It's the smallest naturally occurring piston, really. And what we're pretty sure happens is that protons will bind to this ATP synthase and cause it to rotate, okay, which allows it to catalyze that phosphorylation of ATP. Okay, and this is the summary. If we just want to go through it, the first step in the electron transport chain and chemiosmosis, because they're connected. The electron transport chain is referring to the chain of electrons that are transported down that electronegativity discrepancy, and then chemiosmosis is dealing with the ATP synthase. Okay, so this is kind of summarizing both. We have electrons that are carried to that transport system, and then those electrons can cause that active pumping of protons into the intermembrane space between the two membranes of that mitochondria, and then we have the formation of water, and then that proton gradient causes that rotation of the ATP synthase, which catalyzes the phosphorylation of ADP. Okay, and there's your summary. You're in and you're out. Okay, and this is per 1 glucose molecule. Okay, the discrepancy here, I think we mentioned a little bit later on in the PowerPoint. There's a range, okay, in terms of what yield of ATP that you can get. Also, the other thing to notice is, we mentioned earlier as well, is that NADH can actually form 3 ATP, whereas 1 FADH2 can only form 2 ATP. That's just because of the difference in terms of what these molecules can do structurally to provide energy. Okay. Okay, this is a really good summary, so it's not showing up here, but if you go to the actual PowerPoint itself, these white boxes are removed, and it gives you the summary of the yield at each stage. Okay, of course we have our 2 ATP from glycolysis, then we have our pyruvate oxidation, and also we get NADH from glycolysis. We have pyruvate oxidation that doesn't produce ATP directly, but it does produce NADH, which can go into the ETC. We have Krebs cycle, which produces some GTP, not very much, really just 2 GTP, which corresponds to 2 ATP. Okay, but then we get a lot of these electron carrier molecules from the citric acid cycle, which then translates into quite a bit of ATP in total. Okay. This is just a brief mention in terms of why there's that kind of variability in terms of ATP that are synthesized. It depends on the shuttle. Again, you're not expected to know this, just some people are curious about what that discrepancy is. Depending on what shuttle is being used, you can get variability in terms of the ATP that is produced. Okay, there's something kind of interesting called uncoupling. Sometimes what the protons do is, instead of flowing back down their concentration gradient through ATP synthase, they can flow through these proteins that are called uncoupling proteins. So you know what coupling is. Uncoupling is the opposite. You take the protons, and instead of shuttling them through this, they shuttle through a different transmembrane protein, and that results in some heat being produced, which really is a good thing for a lot of organisms, because that allows, for example, hibernation. Infants, you might have heard, have slightly higher body temperatures than adult humans, because they can't produce a lot of thermal energy through movement, so that they would uncouple their ATP synthase from the protons, okay, and then those protons would flow through the uncoupling proteins.
Mommy, what is that timer?
And those are your questions. So give those a whirl. I'm going to do a quick kind of overview of what I left you to look at on Friday as well, and post that on the class website as well.